So let's start by taking a bit of a refresher on exactly where we're at. Where we left off, we were talking about how DNA is used to synthesize an RNA molecule in a process called transcription. And the purpose of transcription ultimately is to create a protein. So the DNA specifically makes an mRNA molecule inside the nucleus of a cell. The mRNA leaves the nucleus, goes out into the cytoplasm, where the mRNA is used to synthesize a protein. And so what we're going to talk about next, and actually the last thing that we're going to talk about for this class, is how the mRNA ultimately synthesizes the protein. Within the mRNA molecules, we have these segments that are called codons. A codon is a set of 3 nucleotides in an mRNA. And each codon, each set of 3, codes for a specific amino acid. And this sequence of codons in the mRNA molecule is what we refer to as the genetic code. The genetic code is just a sequence of codons in an mRNA. There are 64 total ways that you can put together 3 nucleotide combinations. And your textbook has a great organization, or a table 22.3, that shows you all 64 possible ways that these 4 nucleotides can be combined in 3 nucleotide combinations. So for example, the way the table is laid out, each one of these boxes right here represents a codon. So this is UUU, this is UUC, this is UUA. You get the idea. And then next to that, this is telling you the amino acid that is being coded for by that particular codon. And you'll notice that there's a lot of repeats, meaning that there are looks like there's at least 6 different codons that code for the exact same amino acid. And that should make sense to you because, as I said, there's 64 different ways that we can combine the nucleotides, but there are only 20 amino acids. So there's going to be a lot of repetition here. The way that you can use this table, if you're looking for a particular codon, you can just kind of look for it and find it. Like if we were looking for the AAG codon, you can just kind of look around until you find it. AAG is down here. Or you can use the organization. So it's telling you the very first nucleotide, which you're going to look up right here. So these are all the ones right here that start with A. And then you're going to go to the second nucleotide. And here are all the ones where A is the second nucleotide. And then you're going to go to the third nucleotide, and you're going to find the A's again. And so here's all the third nucleotide is going to be repeated. Right, but you're just going to use this table, whichever way makes the most sense to you in terms of finding the different parts. For me personally, I find it easier to just look until I find it. It doesn't take that long. So you'll notice that some of these do not code for an amino acid. So some of them code for stop, which instructs the mRNA that the protein has finished synthesis. And so the ribosomal RNA, when it reads that, it knows that the protein is done being synthesized. And some amino acids code, or excuse me, some codons code for the start process, which is not indicated as start on this particular table. It's right here. So this is our start. Ooh, did not mean to zoom in there. This is start. So if you're looking at a different table of amino acids, it's going to be laid out exactly the same, but this particular one might say start instead of MET. So let's practice a little bit of looking up and interpreting codons with a worksheet.